Okay, hello there, and welcome back to the Classics of Immunology Journal Club. And before we get started, don't forget to like the video on the YouTube channel and click on the bell to get notified for more. Subscribe to the channel and check out my website, particularly the writing tab, because you'll see all the classic papers of immunology listed there so that you can have access to them and read them. Today, we're going to be talking about a paper from 2004 that was published in the Journal of Immunology. And the authors are Hwang and 5 other people. And then the senior author was Maria Luisa Alegre. And this paper was one of the few, in my opinion, that really shed some light on the whole phenomenon of the regulatory T cell. The title of this paper is "The Transgenic Expression of CTLA-4 Controls Lymphoproliferation in IL-2-Deficient Mice." And in background of this, you'll recall that in 1991, Ivan Horak over in Würzburg, Germany, first made the first IL-2 knockout mouse. And that mouse was interesting because everybody expected it to be immunodeficient, which it was when it grew up after birth. In the first month of life, but in the second month of life started to look not so good. Got a runting disease, not unlike GVH disease, and ultimately succumbed with autoimmune hemolytic anemia on the one hand, and also ulcerative colitis. So that was very, very interesting. Horak published another paper in 95 that after he had transferred the gene, the knockout, onto a BALB-C background from an outbred strain, and that strain of mice got sicker quicker and more severely than the outbred mouse. Then in 1996 and then again in 1998, Marie Luisa Alegre looked into the phenotype of cells that expressed CTLA-4. And as you may recall, the so-called regulatory T cells that Sakaguchi had brought to everyone's attention had several markers that they began being used to check on the phenotype of a regulatory T cell. One of which was a molecule that was called CTLA-4, cytotoxic T lymphate antigen, number 4, that was originally discovered in 1987. And it took them a while before they figured out that, and got different reagents and so forth, that this molecule that was activated on T cells once they were stimulated by the T cell receptor or anti-CD3, for example, they would express the CTLA-4 molecule. And that molecule was inhibitory for T cell activation, proliferation, and function, which was interesting. And so that they ultimately found that this CTLA-4 was in the same family as the co-stimulatory molecule that was given the CD28 designation. In the introduction of this paper, Marie Luisa Alegre points out early on, it's really the IL-2 signals, and that's what her other papers had been about. The IL-2 signals after T cell receptor activation, of course, the cells then are activated to express IL-2 and express IL-2, produce IL-2 and express IL-2 receptors. It's the IL-2 receptor interaction that activates the expression of CTLA-4 in all of the other activated T cells. The thing about this CTLA-4 is that it's constitutively expressed without activation in the so-called regulatory T cell subset. Marie Luisa Alegre wanted to find out what the role of CTLA-4 was in the syndrome that IL-2 knockout mice came down with, since CTLA-4 was an IL-2 induced molecule, gene expression. And in this paper that I'm going to review today, what she found was that CTLA-4 was responsible for decreasing the lymphoid activation and accumulation in the IL-2 knockout mouse. And that whole effect was really on the activation of the effector T cells in the periphery, not on the Treg population. That's the introduction. The data in this paper, I don't want to go through all of it, but just some of it. And so let's do the screen sharing. So this is figure 1. And in the A portion, so figure 1 shows that CTLA-4 expressed as a transgene in T cells is also is equally expressed in IL-2 knockout mouse. So if you look at the A panel here at the top, we have CD4 positive cells, we have CD8 positive cells. In the upper panel, we have the litter-mate controls of the plus minus, the heterozygote of the IL-2 knockout mouse, which is down here in the lower panel. And here's the transgenic CTLA-4 into the heterozygote. And so now we've got 41%. There's no activation now through the TCR. There's just expression, but it's expressed equally well in the IL-2 knockout mouse as it is in the litter-mate control, and as well in the CD8 compartment. Same sort of thing. So in figure 2 then, on the next page, you can see that the CTLA-4 expression prevents the lymphoid accumulation in the IL-2 knockout mouse. So here is what we have at 7 weeks of age and at 16 weeks of age. We're looking at the spleen weight on the ordinate. And what we see here, the wild type, the CTLA-4 transgenic, the IL-2 knockout, and you can see that it's starting to, at 7 weeks, it's starting to not look so good. And the transgenic, the CTLA-4 transgenic brings it down, and it's even more evident at 16 weeks than it was at 7 weeks. And the lower panel basically shows that it's both in CD4 cells and CD8 cells. That's in figure 2. And now if we go on to figure, go all the way on to figure 8 now. And I recommend that you read through this paper and look at all the data very carefully. But I just wanted to show you the most dramatic effects. This is in figure 8, shows the transgenic expression of CTLA-4 prevents the autoimmune hemolytic anemia in the IL-2 knockout mice. And of all the different immunoglobulin isotypes, IgE and Ig gamma 1 are elevated in the IL-2 knockout. But then if you express the CTLA-4, it brings it back down to basically baseline. And then if you look at the hemoglobin concentration in this mice after 16 weeks, you see that the hemoglobin concentration has gone down from 14 grams per deciliter down to about 4 or 5 grams per deciliter, and that's prevented in the knockout. So that's very dramatic. Now the question is, what's going on with the another aspect of this whole syndrome, and that's the ulcerative colitis, because both of these things kill the mice. And you can see here that the CTLA-4 transgenes still get a marked infiltration of lymphocytes into the mucosa. So the ulcerative colitis is not prevented by the CTLA-4 transgene in this IL-2 knockout mouse. So we'll stop the screen share at this stage of the game and go on to the discussion. And as far as I know, that's still, this whole phenomenon of the ulcerative colitis is still not terribly worked out, but there's a lot of other genetic deficiencies that have been created through the knockout system that will cause the mice to come down with ulcerative colitis or colitis in a general sense. The T cell receptor, if you do a T cell receptor knockout, if you do a TGF beta knockout, if you do an interleukin 10 knockout, they all come up with the same syndromes. In the discussion, Marie-Louise enumerates several different points. One is that the CTLA-4 transgenic expression prevents the progressive lymphoaccumulation and hemolytic anemia, but not the ulcerative colitis. And secondly, in normal mice, interleukin 2 is necessary for optimal CTLA-4 expression. And some of the data that I skipped over the effect of the transgenic on Treg population, and they don't restore the Treg population in the IL-2 knockout mouse. And even without the functional Tregs, overexpression of CTLA-4 prevents the increase in lymphocytes and the hemolytic anemia caused by the IL-2 deficiency. So she concludes that CTLA-4 inhibits T cells by several different mechanisms. One is that they scavenge the B7 molecules on the surface of antigen presenting cells from activating CD28, the co-stimulatory molecule on T cells. And that's due to a competitive mechanism. And that was shown by other people to be the CTLA-4 affinity for CD28 is higher than B7 affinity. There's a direct effect of CTLA-4 expression on cells that have CTLA-4 to decrease via inhibition of transcription through the NFAT pathway of activating cytokine genes, particularly interleukin-2. That's it in terms of this particular paper. Now, subsequently, there's several other papers that we're going to get into in our journal club. One is, so that was 2004. In 2007, Pushpapandian and Mike Leonardo from the NIH, in the same group of Ethan Shivak, Bill Paul's group, and because the question was, well, what are these Tregs doing? And a lot of people said, well, they're just a big sink for IL-2. They're sopping it up like you can't believe. And so Pushpapandian, as a postdoc with Mike, she decided that she wanted to look at that. And politically, that was difficult because of Ethan Shivak and others within the Bill Paul lab that were really pushing this whole Treg idea of being a wonderful new subset of T cells that were the answer to everything that anybody wanted to know in immunology. Pushpapandian, in a very nice paper in Nature Medicine, I think, we'll go over it, showed that the Tregs consume interleukin-2, leading to activation or cytokine withdrawal of cell death in the whole population of cells. So not only do they suppress cells, they kill them through cytokine deprivation apoptosis. And that was 07. In 010, Onur Fienerman and Gregoire Altan-Bonnet's lab at the Sloan Kettering collaborated with me, and they basically showed very, very nicely that what happens with the Treg population, subpopulation, is that there is a marked upregulation of the alpha chain, IL-2 receptor alpha chain on the cell surface, which increases the affinity of the IL-2 receptor interaction markedly into the femtomolar range, 10 to the minus 15th molar. And that gives the Treg population a competitive advantage of binding, internalizing, and degrading IL-2. So that sort of goes along with what Pushpapandian had already shown in 2007. So the other thing that is subsequently happened is that everybody was using specific pathogen-free mice, so that they weren't, they weren't dirty mice, but they by no means were germ-free, because it's much more difficult to make germ-free mice. When germ-free mice were finally made and tested in the IL-2 knockout story and so forth, it was found that they didn't get the ulcerative colitis. So there's more to this story, and we're going to be going over all these papers as time goes on. That's it for now. Thanks for listening and watching. And don't forget to like the video and subscribe and click on the bell and check out the other papers. Read this paper very carefully. It's been fun. Thanks for watching. If you're new to immunology, or you just want to delve into a certain aspect of this large field, and you want to get a grasp on things, you should buy my new book, Molecular Immunity, because it's a chronology of all of the important experiments and discoveries that have been made in the last 60 years. It's concise, but it's also very comprehensive. And it's the only such book that's been written since 1970. So you can buy it on Amazon, and together with the YouTube videos of the Classics in Immunology Journal Club, you can't go wrong.